Okay, so what I'm going to talk about is the cancer microenvironment. And hopefully by the end of this, you could see the power of using multiple types of imaging techniques to answer different types of questions. And so this is just an overview of what happens in cancer in relation to the biophysical cues surrounding tumor cells. So what we know is that extracellular matrix alteration, tissue stiffness goes up. That means that around the tumor, it gets very, very stiff. Fibrillar collagens appear. And of course, this actually impacts cancer cell response. So cells start to migrate out, they become more invasive, increases survival and proliferation. So in our work, we use multiple studies. I'm going to show you some highlights of our study using fluorescence lifetime imaging, FLIM, that you've heard of already, to look at cellular metabolism. And what we were interested in is, does the stiffness of the ECM impact metabolic changes in the cell? We also got interested in metabolism in regards to mitochondria. And so what you're seeing here is a cartoon diagram of what we think could be happening at the microscale level. So as the cells start to protrude in the extracellular matrix, the forces go up because there's a stiffening of the collagen here. And we speculate that there could be recruitment of mitochondria that fuels ATP production so that the cells can continuously produce ATP that are needed by a lot of proteins that are there. I'm going to show you one of them called BRAC, they're GTPases, that enable the cells to move through collagen and through that matrix. Why is this important? We know that our lab has been interested in mitochondria for a long time. And what we think that could be happening is exactly what I said. In breast cancer cells and in glioblastoma, I'm going to show you some examples of cells that are in the brain, cells that are in the breast, and how cellular metabolism can be impacted by how stiff the matrix is. Okay, so as you learned, we can excite NADH, which is nicotinamide adenine dinucleotide. It's a small molecule that every single cell in our body has. So if we excite it with 2-photon excitation, we know that we can get some readouts of metabolism. And what is happening in the metabolic state? So just a brief overview in the biology. Our cells naturally make ATP through oxidative phosphorylation. This is the OXPHOS pathway. However, tumor cells and proliferative tissue like stem cells can actually produce ATP whether there's oxygen in the environment or not. Typically, OXPHOS happens when there's oxygen present. When there's a lack of oxygen, glucose gets broken down and makes like 85% lactate. In this case, known as the Warburg effect, whether we have oxygen or not, glucose can be made in the mitochondria and ATP can be produced. So now there's a huge field known as the Warburg effect theory, and there's like 6 different levels of metabolic profiles that cancer cells can adapt to. So what happens during the metastatic phase? The ATP levels go up, biosynthesis goes up, and proliferation increases. So how does the extracellular environment impact cancer cell metabolism? So we went back to some old papers where they showed that if you grow breast cancer carcinoma cells in different stiff materials, this is PAA, and essentially the rigidity increases, what you could see is that these little spots here start to appear. These are invatopodia that start to dig into the matrix. And so what we're showing here is that the stiffening of the environment promotes the cutting of the material. Another thing that happens is that metabolism modulates cancer cell invasion. If we change let's say, the nutrients of the media, this is glucose, this is galactose, this is galactose plus oligomycin, what you see is that MB231 cells, these are breast tumor cells, changing just the nutrients in the media can also cause the matrix to be degraded. So there's a 2 connection here. The first connection is what you saw earlier, where the stiffness of the material, it causes invatopodia to form, and the nutrients can actually alter the degradation process. So can we combine these 2 things using the techniques that you learned this week? So the first thing to do is to grow cells on collagen matrix, right? So this is what we did. This is collagen. This is a gray color scale value here. And what we wanted to know is, can we determine the stiffness, let's say, of the matrix just by images? So we took 2 photon excitation images of collagen. We did image correlation spectroscopy, and I talked about ICS. Paul Wiseman developed this with Niels Peterson. There's no motion here. If we correlate the intensity as a function of space in the X and Y direction, we're able to derive the autocorrelation function that gives us information about the thickness of the fibers. We'll fit this to a 2-component fit model, and what we get is the standard deviation of this, which is the average size of the structure. This is omega naught here. And the amplitude of this function is the signal to background, as well as the proportion of the particular structure. So using this calculation, we're able to see how big those fibers are. So here's now the results. We took collagen, we polymerized the collagen, and 2 different concentrations. The first concentration density was very thick, 3 milligrams per milliliter. And the other concentration was less thick, so more soft, 1.2 megs per milliliter. If we looked at omega naught here, we noticed the fiber thickness is different quantitatively. You can see it by eye that this is like more dense than here. And then the density, which is 1 over the amplitude of the function, the density is also different. We also measured this with rheology experiments where we take the material, we press it down, and we can calculate the modulus of that material. So you could see for sure that 1.2 versus 3.2 megs per milliliter of collagen, and we have 2 G derivatives here from the modulus. You could see that they're statistically significant in terms of stiffness. All right, the reason NADH is important is, and it's fluorescent, is due to its molecular structure. You see these conjugated double bonds here, and these 2 rings are closed in the free NADH form. When a protein binds to this complex, to this small structure, this complex opens up and the lifetime gets much longer. So in this condition, the intensity or the quantum yield is super low. That means that lifetime is very short. When proteins bind to it, the intensity goes way up. The lifetime becomes much longer. So by having a measure of free and bound NADH, we can actually determine changes of metabolism. So I'm going to explain, we already had a FLIM lecture, but we're using pulsed excitation, using 2 photon excitation at 740. The free NADH is 0.4 nanoseconds. The bound is approximately 3.2 nanoseconds. This is bound to LDH. And as Professor Jameson alluded in his talk, if you change the type of protein that it binds to, it could change the lifetime of NADH. So I want to put that in there. So in the lifetime of the phasor lifetime, what we know is that a linear combination of 2 possible states can be just along what we call a trajectory between those 2 points. So if our experimental point falls somewhere in between the 2 possible combinations, we can determine the fractional contribution of one state to another. So in our image here, every single pixel has a fluorescence lifetime, let's say a histogram. And instead of fitting this, we transform this into Fourier space, getting the phasors. And now we can calculate the bound to free ratios of NADH. And in this example, this is it's a spheroid, breast cancer cells grown in a sphere surrounded by fibroblast cells. And you can see that the metabolism, that means the bound NADH here is higher in the normal cells. That means they're undergoing OXPHOS. And these tumor cells are more glycolytic because they're different NADH components. Okay, so let's go back to our experiment where we wanted to change the stiffness of the material. So here we have glass, 3.0 megs per mel, 1.2. So we go from the stiffest, what's the stiffest surface we can have? Is the glass, right? And then we put the cells of collagen, 3 megs, still pretty dense, and 1.2. And what happens is that in MCF10A cells, these are non-tumor cells. These are non-cancerous tumor cells. The metabolism doesn't change. The metabolism is the same. We have high fraction of bound NADH. Now we look at these two columns here. These are ER-PR positive cells. They have a receptor that is positive in these breast cancer cells. These cells are relatively easier to target because they have these receptors. The hardest cells to target are the triple negative breast tumor cells. These are really hard to target with chemotherapy agents because there's no receptors that identify them. So they're more aggressive. And what these more aggressive tumor cells actually have already start in a different metabolic profile. They have a higher free NADH, and as a function of stiffness here, they go from pink to more blue. They go from free NADH to a more bound state. And if we use an inhibitor, this is a Y27632 inhibitor that downregulates the tension of myosin 2. That means that the cells no longer are in like a tension. They can't sense the ECM stiffness. You see that their metabolism is pretty much the same as the soft material. All right, so overall, what is this data telling us? Is that if we grow cells on soft matrices, the oxidative population of the metabolic state goes up. Okay, so just remember that soft material causes these cells to have more bound NADH, and that means OXPHOS is going up. All right, that's what happens in cancer cells, very aggressive breast cancer cells. We also partnered and collaborated with the lab of Stephanie Sedlitz, who's now in UT Austin, and her postdoc, Ali. And what they did is they took the spheroids from glioblastoma patients, they put them in a matrix, stiff matrix in a soft matrix, and they watched those cells as a function of days. And you notice in day 6, in the soft condition, these cells are coming out, they're becoming more invasive compared to the stiff material. Okay, so they are a lab that focuses on RNA-seq information. So genetically, they were trying to find what happens to the metabolism under these two conditions. So they are looking for genes that are either upregulating or downregulating mitochondria-encoded genes. And what you see in the soft material, the mitochondria-encoded genes are downregulated. And the stiff material, the mitochondria-encoded genes are upregulated. So we wanted to match to see if this happens in our conditions in FLIM. So we took their spheres, their cell culture spheres, and these are patient-derived cells. So they actually isolate these from glioblastoma patients, and they put them in stiff and soft conditions. And what we see is that the soft hyaluronic acid, because this is no longer collagen, they have to use a different matrix called hyaluronic acid, actually increases the glycolytic pathway. So the glycolytic pathway goes up. And, you know, we were wondering what the heck, that's opposite of what we saw with the other experiment in the breast cancer cells. And my student who was working with Ali, he said, okay, this whole spheroid has a lot of information. And you see that these cells are coming out after day 6. Why don't we focus on just the tumor itself? And if we zoom in to the regions where the cells are migrating out, do they have different metabolisms? Okay, so that's a very valid question, right? Do tumor cells, as they're migrating out, did they switch metabolism to help them move? And so he zoomed into these regions here, and you can clearly see that the fraction of bound NADH, which are more red, actually goes up. So while in the soft matrices, this appears to be more glycolytic, the cells that migrate out actually are more OXPHOS. So invading cells actually increase their OXPHOS state. So what that's saying is that these particular cells change their metabolism as they're actually going to move out into the environment. So coincidentally, we partnered with Devin Lawson and Dennis Ma, her postdoc, And they're doing these studies where they take primary triple negative breast cancer cells from patients. They put them into mice in local sites. Those cancer cells metastasize to different parts of the body. They go to the lung, the liver, and different regions. Then they isolate the cells from those tissues, and they identify the cells because they have human marker CD268 labeled with mCherry. And they're able to genetically profile using single-cell RNA-seq. And what you see here is all of the proteins that are either upregulating OXPHOS. These are more metabolic markers. So we decided with their lab, we said, okay, we know OXPHOS is going up when the cancer cells migrate and they end up in the tissue. What if we shut down OXPHOS before we put them into the mouse and see what happens? How many cells are found in the liver and the lung and all this? So we use an inhibitor called antimyosin A. And using this particular inhibitor, what we noticed with the FLIM after treatment, the number of hours. So in the control sample, the bound here is more pink, so more OXPHOS. And then we shut down the OXPHOS pathway because we said they're OXPHOS, they're going to go to the liver, they're going to go to the lung, they're going to go to these parts. Let's shut down OXPHOS by using oligomycin. And now we converted them from more bound OXPHOS to more free, more glycolytic, right? They're supposed to be not invasive. And then for sure we have for both cells, patient-derived cells. All right, so when they counted the number of cells in the tissue, I know this is a lot of data, but the oligomycin-treated cells significantly in the number of cells were much lower. So we were able to shut down OXPHOS pathways, less cells went to the tissue, and we have data for both cell types. So treated cells with an inhibitor and they injected back, how much metastasis to go to the site, much less cells. So we were able to switch their metabolic profile. All right, so overall, what you saw so far is that metabolic shifts in cancer progression, that the micrometastatic cells here have a large proportion of gene regulatory proteins that are OXPHOS. That means that they're more prone to migrate and invade the tissue. We can shut down that pathway using inhibitors, and we can verify that with FLIM by switching the metabolism. And then at the primary site, we calculated the FLIM signature, they're more glycolytic, and the metastatic cells are more OXPHOS. Similar in the glioblastoma cells, we have the soft hyaluronic acid glioblastoma multiforme cells. They're also at the core very highly glycolytic, and their invading cells are more OXPHOS. So similar metabolisms as the breast cancer cells. Okay, so now you see how you can look at fluorescence lifetime, autofluorescence. There's no label in any of those cells other than the NADH molecule. So what we are interested in is if OXPHOS is the switch, right, oxidative phosphorylation to invasion, what is happening at the molecular level? So to do this, I will show you some experiments that were done a while back, perhaps students that grew cells on these nanopattern substrate surface material. And we already saw the correlation of stiff material with metabolism, but we also use another biosensor, which I'm going to show you to maybe have some insight as to what's happening. So the first thing that we asked is that if we grow cells on these materials, let's say on nanopattern lines and these posts, does the number of focal adhesions, let's say aggregates change? And I think you heard the lecture of number and brightness already. So the theory was that if the cells can sense the pattern surface, perhaps the way the organization of the focal contacts changes their distribution. And in fact, we did see that on flat surfaces, the ratio to monomers, so we see high order dimers, but in these posts, it goes down. At some point, very far away distances of the nano posts, it varies. But what happens is that the number of population of dimers goes relative to monomers goes down, the number of trimers and more goes down. So what's happening is that the cells are sensing that structure. And they're saying, okay, am I going to attach? If I'm going to attach, how big am I going to make those feet? My feet are going to be very long, or my feet are going to be very small. And with this data, we thought that their feet, as a function of distance in post, actually decreased. So the further the posts were away, the less that those focal adhesions attached. What am I talking about? Here's some movies to show you. Here are cells. We track the cells on flat surface. This is just on glass. Here on posts that have a periodicity 200 nanometers. So the distance between the two amplitudes of the post is 200 nanometers. And clearly what you see is that there's no directionality preference for flat material. But with these posts, what happens is that these cells respond to the nano surface and they actually migrate by turning. Okay. And what happens when we put them on lines, on the line grooves, even if they're 800 nanometer in distance, the cells actually prefer to be elongated and they travel along the nanopost. And here's the rogue cell that doesn't pay attention to the to the pattern and it just keeps going. But that's an interesting cell because then the question is, why is that cell so different, right? Compared to all the other cells that are aligned on the nano pattern surface. So the increase of population of paxillin aggregates decreases the migration length. So we calculated the speed of how fast the cells were moving. So as the protein, and I didn't have time to explain this, what's happening with the paxillin protein is that smaller aggregates are disassembling when the cells have to move. So it gets stuck. Now those proteins disassemble, it goes forward, they reappear, then they dissipate, and they continue to do this. But the aggregates get smaller and smaller. And they get smaller and smaller as the distance between the posts gets shorter and shorter and shorter. Okay, so there's definitely a trend here. Okay, so can nanotopography modulate focal adhesion turnover? We did see that on the smallest nanopillars, they are the most motile. So the answer is yes. And the aggregation state decreases with decreasing nanopillar size. So as the distance between the nanopillars as it gets smaller and smaller, the focal adhesions disassemble as smaller and smaller aggregates. So increasing aggregation disassembly results into a greater migration. So we're interested in mitochondria, and there's a lot of work there saying that mitochondria translocation in cancer cells is important. So there is a protein called synaptophyllin. Synaptophyllin can be shut down using siRNA. And what happens is if you shut down synaptophyllin, the cells cannot move very far from the origin. So this is traces of cells moving from this origin. And you could see that with synaptophyllin siRNA, you shut down synaptophyllin because it's a stop for the mitochondria, cannot go anywhere. Then the cells can migrate everywhere. So what this is saying is that less synaptophyllin means faster mitochondria translocation and cells travel further. So in order to test this theory, we wanted to make the cells move. And to make the cells move, you have to wait a long time. And my student didn't want to sit there and wait for hours to see the cell move. The movies I showed you, the cell moving, took 24 hours. So she said, I don't want to do this. So I told her, okay, we have this sensor, this RAC protein that enables the migration of cells to happen much faster. So what you saw here is if I photoactivate this sensor, this is a LOVE protein that's sensitive to 405 nanometer light, it comes off, RAC becomes activated, and as you move the laser, you're activating RAC, and the cell follows the laser because that's the direction where RAC is going to extend and protrude. So it's almost like artificially making the cell move in a given direction. So we said, okay, let's use RAC. And now in these movies, these are MB231 cells. These are the very aggressive breast tumor cells. These are the non-aggressive, they're not tumor cells. And we labeled the red is RAC and the green is mitochondria. And so we asked, if we photoactivate RAC, will we see preferential recruitment of mitochondria to the edge? Here, and I have to apologize, this was very close to the mitochondria. So later on, my student moved this. So I'm going to play one movie at a time. You know why it's playing so slowly, but maybe that's a good thing. So far, there's one teeny tiny mitochondria being recruited. And then as a function of time, the mitochondria starts to migrate to that region of active recruitment of RAC. Okay, and then if I play this movie on the bottom, I don't know if it's going to play. We don't see any active mitochondria recruitment here. And then I told my student, you're too close to here. Why don't you move the beam? So the edge of the cell is here. And then she's going to move it, and she moves it here. And then we don't see too much recruitment. We don't see any recruitment of mitochondria there. So we did perform a lot of experiments, and we watched the recruitment over a function of time, and we did them on NIH3T3 cells, MCF10A. These are two control cell lines and the aggressive tumors. We use a directionality ratio because the mitochondria go like this in all directions. They don't go just straight. So we had to calculate the deviation from a straight line. And you could see that using photoactivatable RAC, when we photoactivatable RAC, we had higher directionality. If we don't photoactivate RAC, we still get a high directionality ratio. And this is just the control with nothing. We just don't do anything. And then the average speed of the mitochondria also correlates with the photoactivation of RAC. So mitochondria are really important. And do Lionel stop me if I'm going over time? But we wanted to understand, can we track mitochondria and look at the dynamics of mitochondria and see if we can actually do FLIM on the mitochondria when cells are moving. So when a new student took over this project, he saw that in all of the literature, they were using Fiji to track mitochondria. And if any of you have ever used like Fiji or ImageJ or something to track, let's say, a vesicle, you have to do one click at a time. And he said, forget this. I don't want to spend my life clicking thousands and thousands of images to do this. And so there's a lot of literature using Fiji and other tools. And then the problem was it's hard to distinguish mitochondria from that image. You can't barely identify, is this one, is this two, is this three? So, and they're overlapped, right? So you have to find, you can go up to 10,000 clicks just for one condition, and it's biased. And it's user dependent. So my student said, okay, I'm gonna write a new algorithm to trace and track mitochondria. So this is the original image, and he looked at this image and he said, there's too much background here. I cannot identify single mitochondria. So using astrophysics techniques to remove, let's say, background, he used the diffuse background subtraction algorithm. And so he calculated what is the diffuse background from these images, from the stack. So he got this image here. He subtracted the background. So this is what the background looks like. He Gaussian filtered the image. And then he thresholded, and then the final image that you could see here is isolated mitochondria as best as he could get. Okay, so now we have a final image, and now we can analyze this as a function of time. So he did this for each individual frame. And I don't have time to go into the details of the algorithm, but it's all published in our paper here. By using a minimization algorithm and like a loop iteration calculation, he's able to subtract the diffuse background, and let's say if you have two regions, and this region is a lot of background, but if you subtract the diffused background from here, you get isolated mitochondria, and that's the diffused background from the original image. You use a low diffuse background subtraction from this region, and by using a low diffuse background subtraction, you're able to identify structures that were not visible because of the background here. So just to give you an idea. Using the time frame of all of the movies that you saw, we can calculate fusion and fission events because we can see if the mitochondria was a single object before and after. If it was a single object and then it became two objects, it underwent a fission event. And then, the opposite was true for fusion. We can also look at the mitochondria mean square displacement, so calculate whether it's directed motion or it's just diffusive motion, and get the speed and directionality and the branching of the mitochondria, so how much it's branched. And he took it one more step and he said, why don't we use the autofluorescence of mitochondria to see the lifetime signature? So the problem with FLIM is that you have to acquire enough frames to get a good signal. And the problem is mitochondria is moving. You're not going to get a very good lifetime average of the motion, right? We just get the FLIM signature from that mitochondria, and it's okay if it moves because we're going to integrate the lifetime of all of these pixels from all the frames as it moved. So you get one lifetime data point, even if the mitochondria is moving, and the lifetime is calculated in just this plane. So he isolated the mitochondria, and this is the fraction of bound NADH mitochondria. And he did three tests. The control, which is the non-tumor cells, the ER/PR positive tumor cells, and the triple negative breast tumor cells. Relative to each other, the number of mitochondria are different. The number of mitochondria and the more aggressive tumor cells are very, very high compared to the control. If we look at the cancer fraction of bound NADH, we see that cancer cells, mitochondria are more OXPHOS, they're more bound NADH. And using and look at morphological and motility parameters in respect to bound NADH. And what we see basically is the fact that cancer cells are modulating metabolism, and as a consequence, they carry these processes. Their average, the triple negative breast tumor cells here have a high correlation for speed, but they have an let's say, feature for intensity, directionality, and solidity. Okay, relative to bound NADH. So I think I'm going to speed this up because I feel like I am out of time. So also my student said, well, looking at cells in 2D is not enough. Let's go to 3 dimensions. Can we track mitochondria in 3D? And he did, using patient-derived mitochondria. So these are normal epithelial cells. These are patients that have breast reduction surgery. They're not tumors. They're just breast cancer cells. These are isolated cells, ER/PR positive cancer cells. We have MB231 cells and other triple negative breast tumor cells. And these are xenograft cells. So we did 3D spheroid conditions versus 2D spheroid conditions. And this is a lot of data, but I'm going to summarize here. What's the difference between 3D and 2D? That in cancer cells, the mitochondria is slower, but they're more directional. So there's definitely directionality. They have less burst of speed. It matches the cell migration in 3D versus 2D. Triple negative breast tumor cells undergo more fusion. Fusion enhances cell invasion. Triple negative versus ER/PR positive. What's the difference between this? The solidity, the branching of the mitochondria, not just is it smooth like this, like what you see in textbook, but they have different branching. They're more networked. They have lower membrane potential, and they're less rounded. So in general, breast tumor cancer cells look very similar to normal cells. That's what we concluded with this. But certainly there's a lot more work because my student didn't look at populations that were actually migrating cells. right? So there's a lot of work still left to be done. We can use like a random forest classification system to identify what features are the most important for identifying aggressive cancer cells. And this is just a quick test looking at motility features, morphology features, or combinations. So what features are the most important to tell us these cells are going to be more aggressive? And we can do that also in glioblastoma cells. This is the movies of mitochondria in the 3D culture of spheroids in the hyaluronic acid. So when we looked at this data, we see that the cells in the soft matrices, they travel further and they're more directional. They're less branched and they're longer compared to the stiff material. So soft. Remember that soft matrices are highly glycolytic, stiff matrices are more oxphos. So finally, we're starting to understand the mechanosensing part of these cancer cells migrating and why mitochondria is important. And just in conclusion, you can use multiple tools. Now you have a tool. You go back to your lab, and you don't have to stick to one toolbox. You can do many to understand a very complex biological system. And we're still working on some of these things to try to understand what happens in mitochondria. But I just want to thank all the lab, everyone who worked on the project, including my student Austin, who's now at Calico, and Lorenzo Scipioni, Julia Tedeschi, and everyone at the LFD, and all of our collaborators. And thank you so much for listening.